guys, and welcome to IV Online. Javed Afridi here, and you are watching AS Level Biology. We are studying genetic control, and now it's the time to discuss about the second stage of protein synthesis, and that is translation. So, in the previous video, I told you how transcription takes place and what's going on in transcription. We have synthesized one molecule of messenger RNA from copying a segment of one strand of the DNA called gene. So the information in one piece of DNA molecule have been transferred in the form of messenger RNA having a transcript of DNA. And this messenger RNA now move out from the nucleus to the nuclear pore and reach the ribosome in the cytoplasm, where the process of translation is going to begin. Now, there are three different characters which we have to study. Number one, what's going on with the messenger RNA which have been moved out from the nucleus into the ribosome. So the first character is the mRNA having specific codon according to the process of transcription. The second structure is the tRNA molecule, transfer RNA, which is also another important nucleic acid molecule, tRNA, which is present in the cytoplasm, and it carries the amino acids to assemble them in the formation of a polypeptide. As initially I told you that the messenger RNA is used as a transcript, as a template for the synthesis of a primary protein. So the sequence of nucleotide in the RNA, which is called codon, will be used for the sequence of amino acid. And this sequence will be set by the tRNA carrying amino acid molecule according to the information delivered by the messenger RNA. The third character is the ribosome, which is the machinery that the ribosome actually is the site of attachment for the messenger RNA. It arrives and attaches the messenger RNA. And the ribosome is not only the site of attachment for the messenger RNA, but it's also a site for the transfer RNA. So at one side, the messenger RNA is located, and on the other side, the tRNA carrying the respective amino acids to the messenger RNA following the codon sequence and the formation of peptide bond. So this is very interesting story. And now we will discuss one by one all these three characters. So I am going to start from the ribosomes, from the transfer RNA first. Let's talk about the structure of tRNA and some important aspects of the transfer RNA. So what kind of structures in the transfer RNA we have? You can see the molecule of tRNA over here. This is overall is the tRNA structure, and you can see although it's a single stranded structure, but since it makes loops, and inside the loops we have specific base pairing like the DNA molecule. Unlike DNA, it is a single stranded structure like a leaf of clover plant here, and they have the hydrogen bonding because of their loopy structures. Otherwise it is as a single strand, just not a double stranded structure. They have the base pairing, and in the base pairings no thymine is present. We have just uracil. Now in the entire structure there are two very important area if you have to focus here. Like one of the area which have to be focused is that area which is known as to be the anticodon, and the other one is that area where the amino acid is going to be attached, and this is usually known as the CCA site. CCA site is the site of attachment for the amino acid to be here, and this is called anticodon. So this site is used to interpret the messenger RNA. Let me to delete this one for some right. So here, sorry. Yes, now it's clear. Here in this structure, you can see that the two important structures in the tRNA, three important things. Number one, tRNA is a single-stranded molecule like the messenger RNA. However, unlike messenger RNA, it is a loopy structure, so it has specific base pairing. There's no thymine inside them. And there are two important areas. One is the terminal site here. You can see the terminal site where we have three unpaired bases. You can see the uracil, guanine, and guanine, but there's no hydrogen bonding. There's no other pair with them. So this will be paired with the nucleotide on the messenger RNA. So if there's a messenger RNA and they have the sequence here as adenine and cytosine and cytosine, so that these three can be paired up with this messenger RNA molecule, as they have U against A, G against C, and G against C again. So this site is the attachment for the messenger RNA. And this is the sites which is used for the attachment of amino acid molecule. Each amino acid molecule follow the anticodon. So anticodon is complementary to codon. Codon is complementary to genetic code. So if we have a genetic code on a DNA strand TAG, this is one of the strand on the DNA. So its codon on messenger RNA will be AUC. So AUC, this is on the non-transcribed strand or the coding strand. And similarly, its anticodon on the tRNA should be again UAG. So against A we have U, where there's no thiamine in RNA, and U against A and C against G. So, all these three will be coded for single same amino acid, but the code of amino acid on the tRNA should be UAG, on the messenger RNA should be AUC, and on the DNA it should be TAG. However, the amino acid, these all three are the code for the amino acid. However, this amino acid is directly interpreted by the anticodon, because you can see here it is attached to the tRNA. Amino acid makes a bond with the tRNA, not with the messenger RNA, not with the DNA. So these codes are far away from the amino acid. However, this code is quite closer to the amino acid, because the amino acid bind to the CCA site of the tRNA, and which is on the other side we have the anticodon. So this tRNA interpret amino acid. So it means like if we have UGG here, so they must have a specific amino acid which should be bound to the tRNA molecule. Keep in mind, this is not attached earlier. The tRNA molecule is not like in this way. It is right like in this way. This site is remain empty. There is nothing to be attached, but once it gets any messenger RNA here, like this is the messenger RNA which has the sequence of A, C, G. So it must be here as U and G and then C. So following U, G, C, there's a lot of amino acids we have. All these are the amino acids. We have 20 amino acids. But any one of the amino acid which follow the code directly as UGC here, this code UGC. So suppose if this one is the responsible for UGC, so that this will become activated and will come and attach to the amino acids, to the tRNA here. So each amino acid have a specific anticodon which is located on the tRNA, and once the tRNA become activated using its anticodon to messenger RNA, it will attract the respective amino acids on the CCA site. This is what about is given here in the transfer RNA. If you see here, so we see transfer RNA translates the message within the nucleotide sequence of messenger RNA to a specific amino acid sequence. These sequences of amino acids are joined together to form a polypeptide or a protein. Transfer RNA is shaped like a clover leaf plant. If you see the diagram here, it's like the clover leaf plant shape. And within three loops here, it contains an amino acid attachment site called CCA site. This is actually CCA site. This is actually CCA, because we also have non-specific base pairing here, unpaired bases. You can see all these are paired, but this part is unpaired. Similarly, the unpaired part is very important. Unpaired. This is also unpaired. So two unpaired bases, one is the CCA site on one end and a special section in the middle loop called the anticodon terminal site here. The anticodon recognizes a specific area on messenger RNA called a codon. So if, like I'm just showing here, you can see here, this is the ribosome, this is the smaller subunit, and this is the larger subunit, and you can see that this is the this is the ribosomes, here is the messenger RNA, and here we have the transfer RNA. So in the transfer RNA, you can see the amino acid is attached here, which is the methionine, and the anticodon is UAC, but this UAC attached to against the AUG only. Once the messenger RNA arrives, it attaches to the ribosome. It's the more clear picture here. If you see here, this is the structure of the ribosomes, and in the ribosome, this one is the larger subunit, and this is the smaller subunit, and the messenger RNA is just sandwiched in between the two smaller and larger bases, and you can see the AUG, so the anticodon should be UAC, then UUC, so here's the AAG, and then CGA, so here will be GCU on the anticodon. So this anticodon code for the arginine, this anticodon code for the phenylalanine, and this code for the methionine. So in this way, the anticodon corresponds to the amino acids, but the anticodon corresponds to the codon, and the codon corresponds to the genetic code on the DNA. That's why I'm just going to tell you once again here in the background. So the anticodon recognizes a specific area on messenger RNA called a codon. The bond between the CC site of tRNA and amino acid is called amino-acyl bond, which is a covalent bond. Here we have a bond between the DNA. This is the part of the RNA, and this is the part of the amino acid. This is amino acid. So the bond between the amino acid, this purple area, and this green area, the bond between these two is called amino-acyl bond. It means that the amino acid molecule is attached to the tRNA, which is activated by amino-acyl bond. Since there are 20 amino acids essential for our body protein synthesis, there are minimum 20 different kinds of tRNA. You know that we have 20 types of 20 types of amino acids. So if one amino acid is going to attach to one tRNA, it means we have at least 20 different types of tRNA, each having a specific amino acids. However, here we have the anticodon, and anticodon are complementary to codon. And I told you that we have 64 genetic code. Initially, I told you, so if we have 64 genetic codon, we must have 64 codon as well. If there are 64 codon, we must have 64 anticodon as well. They are just opposite to one another. So if there are 60 anticodon, it means we have a maximum of 64 different types of tRNA. So we have a maximum number of tRNA on the basis of anticodon, which is 64, and we have minimum number of tRNA on the basis of amino acids. We have 20 because we have only 20 amino acids. So 20 amino acids are coded by 64 different genetic code. It means that different tRNA, sometime 3, sometime 4, even 6 types of tRNA can carry the same amino acid, because the code is degenerative, I told you in the previous lecture. So this amino acid, if attached to, suppose this amino acid is alanine, suppose, and it shows the code as UGG. So if there is another tRNA, suppose here, and it has a different code like UCC, so might be possible that it again have the same alanine, because UCC and UGG both are responsible to code alanine. Not the same code, but I'm just assuming. So it means that two different tRNA, this is tRNA 1, this is tRNA 2. Two different tRNA can represent the same amino acid, in this case, this one and this one. So that's why the maximum number of tRNA are 64 on the basis of anticodon, and the minimum number of tRNA on the basis of amino acids are 20. So they say since there are 20 amino acids essential for body protein synthesis, therefore there are minimum 20 different kinds of tRNA. On the other hand, there are 64 different types of anticodons. So the maximum number of tRNA is 64 on the basis of terminal sites where different anticodons remains attached. So this is the site of anticodon. That's why an anticodon are of 64 types. So we have 64 types of tRNA on the basis of tRNA. And here is the site for amino acids. And we have a maximum number of 20 amino acids. So tRNA should be 20 different types. So we say tRNA may be 20 types or tRNA may be 64 types. But even you are saying 20 types, then you're talking on the basis of amino acids attached to the CCA site. And if you're talking about the 64 types of tRNA, then you are talking in the sense of anticodon, which is located on the terminal site. This is the story of the transfer RNA. Now, what about the next structure? The next structure is the ribosome. So let's see the story of ribosome here. This is the structure of ribosome. And ribosome is made up of two subunits. One is called the larger subunit, the yellow one, which I have shown here. This is the larger subunit. And the smaller subunit shown by the green, light blue here. This is the smaller subunit. Now, in the larger subunit, we have two active sites. Generally, three active sites, but in your syllabus, we usually mention about these two sites. This is the P site, and this is the A site. So two sites on the larger subunit of ribosome. So initially, what's going on? Let me just copy it. Let's make another one. This is the smaller subunit of ribosome. Here is the smaller subunit. And now the messenger RNA arrives here. Let me show the messenger RNA by some different color, by the green one. So here we have the messenger RNA just opened from the nuclear pore, from the nucleus. Here is the nucleus. This is the nucleus. And the tRNA, the messenger RNA has been released. This is the messenger RNA. So this is the mRNA. Copied a segment of the DNA molecule. And here we have the sequence of nucleotide. Let me show the sequence of nucleotide is adenine, uracil, guanine, cytosine, uracil, guanine, uracil, adenine, cytosine, guanine, and adenine. So we have four codes over here, four codon on the messenger RNA. They copied a single strand of the DNA. This is the third site, and this is the fifth one. Oops, it should be from the fifth to third, so let me do the correction. Here, this one is the fifth one, and this one is the third one, because it grows from the fifth side and attached to the start from the fifth side and growing toward the third side. Now, this messenger RNA is going to be attached at the ribosomes, but it attached to the ribosome usually at its fifth site. So the fifth site is, let me move this messenger RNA from here. It should be attached like in this way. You can see these ribosomal structures. So they attach its fifth site to the smaller subunit of ribosomes in such a way that two codon can be in the area of the smaller subunit of ribosome. So you can see that this one and this one, both are inside the smaller subunit of the ribosome. So we can see that now it's attached in the messenger RNA molecule AUG. Let me just show some more elaborate structure again here. I'm just going to copy this structure. Oops. It should be. Okay. Smaller subunit of ribosome. And here we have the messenger RNA to be attached here. This is the messenger RNA. To show it is used in a double line or more prominent. So here we have the sequence of nucleotide now. Here we have the AUG and then the CU and U. So we have 6 bases attached to the smaller subunit of ribosomes. I mean, 3 codons we can attach here. Now, once they attached, the larger subunit is going to be arrived here. So here is the larger subunit. So if I just put like, makes a copy here and paste, so it should be attached like this way. So larger subunit is also arrived here. We attach like this way. You can see here in the same format, like, yeah, no, it's fine. So you can see that it's attached in this way. Let me to delete this structure. Right. Now you can see that the larger subunit is arrived on the smaller subunit, exposing its P site and its A site. Now you can see that the other bases here is the guanine, uracil, and adenine, and here we have the cytosine, guanine, and adenine. This is suppose is the stop code, and this is the start code initially to start the process of translation. Now here we have different amino acid molecule in the cytoplasm. So we have we have the amino acids cysteine, we have the amino acids glutamic acid, we have alanine, we have phenylalanine, we have tryptophan, and so on. And like we can see the valine and so on. And all of these have specific code. Let's suppose that the code AUG here is used for the methionine. So here is the code AUG. The CUU code is used for the alanine. CUU. It's not confirmed. I don't know, because we are not supposed to keep in mind all these code. Just like for your understanding, I'm just assuming these code. Otherwise, some other code may be used for the alanine. It's not the exact code. Just for explaining, just for giving the idea. And the other code is for valine is, let's suppose, is GUA, and the cysteine has the code as G. Sorry, it's C, G, and A. So according to this pattern, this is in the cytoplasm, we have these codes specific for the amino acids. Once the ribosome settle over here on the messenger RNA, this is the larger subunit, this is a smaller subunit, sandwiching the messenger RNA inside, and the two code now inside in the ribosome, larger and smaller subunit. The first code is AUG. Now in AUG, the complementary strand for the AUG, the complementary nucleotide for the AUG should be I'm talking about this one. For these three, we have a complementary code as UAC. And let's suppose that UAC code is used for methionine. Sorry, it's UAC. So we need the tRNA which have the anticodon on its terminal side as UAC, because we have the first one as AUG here. So for its specific base pairing, we require the anticodon should be UAC. So this tRNA must have a code as UAC to bring the corresponding amino acid. So if we just copy over here, let me copy this on the next page. Yeah, so here we have, right. Now look at the story here. We have the tRNA now, and this tRNA have the anticodon UAC. But once we see where is this code UAC, which kind of amino acid should be activated, if we see the code here, so UAC code is used for methionine. This is used for methionine. You can see the UAC code is used for methionine. So in all these amino acids, only methionine will be activated and will be arrived on the CCA side of the tRNA to attach with it. So it means that methionine used by meth, it becomes activated and will be arrived and bound to the CCA site like in this way. This is the amino acid meth. This tRNA now will start moving to arrive on the P site here, and it will share its nucleotide with the corresponding sequence of nucleotide. Similarly, the other code, which is now CUU, you can see the other code is CUU. So two tRNA at a time we require now. Now there is another tRNA, and it has the anticodon, which is against the CUU, must be G, G, A, and A. So if we see the code for GAA here, we see that it is used for cysteine. You can see here the code is used for cysteine. So cysteine is going to be attached here now. Here, cysteine will be attached because the cysteine is going to be activated in the rest of the amino acid. Only cysteine will be activated, and it will make amino acyl bond here, amino acyl bond to attach with the CCA side of the nucleotide. So this is now carry forward here. It will be moving here and it will be attached like this way. So two tRNA molecules now attached to the ribosomes. Once they makes the hydrogen bond between them, specific base pairing, then also a peptide bond is formed between them. So a peptide bond should be shown here by a red color. Here the peptide bond. This is a peptide bond to be formed between the two amino acid. One is methionine and one is cysteine. Once the bond between them two has been formed and the hydrogen bonds also form between them, then what's going on that the bonds between the first anticodon and codon on the messenger RNA on the P side, like on that side, these bonds should be broken down. And these bonds should be broken down, and also the amino acyl bond should be broken down. Once the amino acyl bond should be broken down, now the situation will be like this one. The bond has become broken down, and also this bond is broken down. They just have the unpaired bases again. And now this DNA molecule will leave the active, will leave the P site. The P site will become empty now. Sorry, the P site will become empty now. And the ribosomes, so it remains open now. So now the P site is remain open and the A site have been occupied. Now, once this occurs, the whole ribosome is now moving one step ahead. It's moving front. So when it's moving one step ahead, like in this way, now what's going on that the bond between the tRNA anticodon and the messenger RNA codon at the A site will also be broken down, like in this way. These bonds will also be broken down, and the entire messenger RNA will be shifted from the A site toward the P site. I'm sorry. Let me just show through this diagram here. You can see here. First, it's attached the methionine, and then it's moving ahead like in this way. So the other code, if you see, is the UUNC, and against the UUNC we have the anticodon as AAG, which carrying the amino acid phenylalanine, the methionine on the P side, and this is the A side. They makes the peptide bond in between them. The bond between them is broken down. It's leave the tRNA, and then the ribosomes move one step forward to exposing the next codon, which is the CGA. For CGA, then another one which is the GCU, and attached to the arginine become the activated amino acid, and the same procedure is repeated. So let's now in detail, I'm just going to discuss with some detail the translation process. So what is going on in translation? Stages of translation: chain initiation. Messenger RNA with specific base sequence codon binds to the smaller subunit of ribosomes by using its 5' primer end. As I told you, it is attached to the fifth side. I mean, this is the fifth side and it's the third side. So you can see it is attached on the fifth side, while the remaining side is just outside this ribosome. So on ribosome, it just attached the triplet code triplet codon. Two codon can be attached to the ribosomes from the fifth side, and the remaining part is outside the ribosome. Right. The larger subunit have two binding sites for transfer RNA, P site and A site. When the small ribosomal subunit attached to a messenger RNA molecule, the larger subunit also join the newly formed complex, as I told you here. So once it's attached to the smaller subunit, then the larger subunit also join it, and now they become sandwich the messenger RNA in between the smaller and larger subunit. When the smaller, right? Now, an initiator tRNA molecule recognizes the specific codon sequence on the messenger RNA molecule. Initiator means this is the first code, or the first codon on the messenger RNA will be identified by the initiator tRNA. So since it is AUG, so our first tRNA must have an anticodon of UAC. So for this one, UAC, and for the other one, which is CUU, for that one we need GAA. So the first two tRNA we need, one is the UAC and the other one is the GAA. So two consecutive tRNA we require to bind with the messenger RNA. One has the anticodon UAC and the other has anticodon GAA. Of course, these two anticodon will also attract their amino acids on their CCA side, will become activated, and they will also arrive on their CCA side to form amino acyl bond. So here we see, right, and bind to it through the hydrogen bonding using specific base pairing rule. So the terminal anticodon will bind with the hydrogen, with the messenger RNA on the through the hydrogen bond using specific base pairing rule. The CCA site of this tRNA bind specific amino acids molecule accordingly following the codon on messenger RNA. As I told you, the codon recognizes, the anticodon recognizes the codon, and the amino acid actually following the anticodon. So once the anticodon is attached to codon on the messenger RNA, then the amino acid will become activated and will attach on the same tRNA accordingly using amino acyl bond, through amino acyl bond. The initiator tRNA resides in one binding site of the ribosomes called the P site. When a new tRNA molecule recognizes the next codon sequence on the messenger RNA, it attaches to the open A site. A peptide bond forms connecting the amino acids of the tRNA in the P site to the amino acid of the tRNA in the A binding site. This peptide bond formation is catalyzed by an enzyme called, it's very important to understand, to memorize, peptidyl transferase. So the peptide bond which is formed between this bond I'm talking about, this bond which is formed between the two amino acid molecule in the process of translation is catalyzed by the enzyme peptidyl transferase. And this bond formation where the amino acids molecule are attached to the DNA, to the tRNA, is catalyzed by the enzyme amino acylase. So amino acylase catalyze the formation of amino acyl bond. I told you, amino acyl bond form here, and the peptidyl transferase catalyzing the peptide bond between the tRNA. So the two sites now. Now what's going on next? Chain elongation. This is the chain initiation, how the polypeptide chain is going to be started following the starting code initiator DNA, tRNA. As the ribosome moves along the messenger RNA molecule. Now the ribosomes move one step ahead. How? One step ahead means, like here. can see here we have, you can see here, in this case, the tRNA is attached to this anticodon, to this codon, sorry, AUG, and the anticodon is UAC, which are tracking the amino acids methionine on the CCA side. Now, this ribosome is actually moving one step ahead. So when it moves one step ahead, it leaves the AUG outside, and the UUC will be now inside. UUC will be at the place of AUG, and the other one, which is the CGA, now will be inside the ribosomes. And you can see here, so AUG, and now another one arrive here, the anticodon. This is the previous one, and this is the newly arriving, and it makes the peptide bond in between them. And then you can see that once they makes the peptide bond between them, this bond will be broken down. It remains empty, leaving the methionine and phenylalanine here. Another new one, it will attach here, and the polypeptide formation will remain continue in the same way. Here I'm showing some more steps of translation. You can see here is the messenger RNA, which is moving outside the nucleus. Here we have the ribosomes, and here we have the different types of tRNA. This is the tRNA, and this is the anticodon. Amino acid site attachment, which is the CCA site. Here the amino acid lysine, tyrosine, methionine. There are two different kinds of amino acids, each one following the anticodon to be attached there. So methionine and tyrosine, these two are attached here, but the lysine is not attached here. Why? Because the lysine don't have the specific anticodon on the tRNA molecule. So this tRNA has an attachment site specific for methionine and has an anticodon UAC. You know that UAC is actually the anticodon which is used for the methionine to be attracted and attach it to the CCA site. So here is the UAC. Similarly, tyrosine, this tRNA has an attachment site specific for tyrosine. Why? Because it has an anticodon as AUG. So here we have the AUG. This AUG attract tyrosine, and you know this UAC actually attract methionine. So the amino acid code is following the anticodon, and the anticodon is following the codon on the messenger RNA. So tRNA molecules bind with their specific amino acids. Now this tRNA has an attachment site. So they are, and these are the free tRNA molecule you can see. Now what's going on in the cytoplasm? The messenger RNA attaches to a ribosome. This is the larger subunit. This is the smaller subunit. And you know, this is the fifth site and this is the third site. The fifth site is attached with the messenger RNA. And here is the codon. Here is the anticodon. And this anticodon, this codon is attract the tRNA accordingly, following specific base pairing rule. This anticodon for attracting the amino acid to be attached on the CCA site. And this is the start code actually. So the start code is, I told you initially that start and methionine both have the same code. So the start code is UCA and is the methionine to be attached here. Now, each tRNA has the complementary anticodon to the codon on the messenger RNA. The first tRNA, which always carries the methionine amino acid, forms hydrogen bonds with the first or start codon AUG on the messenger RNA. Keep in mind that each messenger RNA has its first code to be, AUG, which is the start code of each gene, starting one. And of course, the amino acid methionine will follow the anticodon, which is the UAC. So UAC is the anticodon, AUG is the codon, and the amino acid is the methionine to be followed here. Now, what's going on in the next step? You can see that another tRNA is arriving here on the A side. This is on the P side, and this is on the A site to be attached here. Now here, the second tRNA bringing the second amino acids bond with the next codon on the messenger RNA accordingly, specific base pairing, and then a peptide bond is formed between the two. So a peptide bond formation between the amino acid catalyzed by peptidal transferase. Peptidal transferase, the enzyme which catalyzes the formation of this bond, peptide bond. Now, the ribosomes move along the messenger RNA in a 5 to 3. Since this is the fifth one and this is the 3 one, so ribosomes is moving like in this way. It moves one step ahead. So when it's moved one step ahead, this codon will be outside and this codon now will be inside, and another tRNA will be arriving here, like in this way. You can see in the 7th step, the third tRNA carrying the third amino acid bond with the complementary codon. Now this is outside, and its tRNA also breaks the hydrogen bond, leaving its amino acids methionine. So both the amino acyl bond and both the hydrogen bonds have been broken down. The tRNA now free to move away, leaving its amino acids methionine at the ribosome. Similarly, the process remains continued. The ribosomes move one step ahead. Now the other codon should also be outside. Also, again, the peptidal transferase, again, the amino acids, the amino acyl bond and the hydrogen bond will be broken down, and the chain initiation will be remain continued. So the first tRNA now without an amino acid is released. Now, the ribosomes continue to read the messenger RNA molecule, building a polypeptide chain until it reaches a stop codon. So finally, this is the stop code, and the process will be remain continue. Arrival, departure, arrival, departure. So tRNA is leaving, is leaving from here, and they're just releasing their amino acids in the form of a chain formation. So how many tRNA have gone? One from here, one from here, one from here, one. Up till now, 4 tRNA already have gone, leaving their amino acids molecule in the chain. And now the 5th tRNA should also be leaving, and the 6th one, and then the chain will be released as a polypeptide following the code on the messenger RNA and tRNA codon. The ribosomes continue to read the messenger RNA molecule until a polypeptide chain, until it reaches a stop codon. So that's the story of translation. Now, one by one, all these steps to be discussed now. Look at this. What does it say? This stage of protein synthesis occurs in the cytoplasm of the cell. Unlike transcription, which was taking place in the nucleus, after leaving the nucleus, the messenger RNA molecule attaches to a ribosome. In the cytoplasm, there are free molecule of tRNA. These tRNA molecules have a triplet unpaired bases at one end, known as the anticodon, I told you, and a region where a specific amino acid can attach at the other, which is called CCA site. There are at least 20 different tRNA molecules, each with a specific anticodon and specific amino acid binding site. However, if you're talking about the specific tRNA sites, the specific anticodon, then we have 64 types. The tRNA molecule bind with their specific amino acid also in the cytoplasm and bring them to messenger RNA molecule on the ribosome. So they just pick up the amino acids. They just pick up the amino acid from the cytoplasm, attach it to the CCA site, and then carry it toward the messenger RNA codon specific, following specific base pairing rule. The anticodon will match with the codon, and the amino acid will be brought to the ribosomes. The triplet of bases, anticodon, on each tRNA molecule pairs with a complementary triplet codon on the messenger RNA molecule. 2 tRNA molecules fit onto the ribosomes at any one time, bringing the amino acids they are each carrying side by side. A peptide is then formed between the 2 amino acids molecule. This process continues until a stop codon on the messenger RNA molecule is reached. This acts as a signal for translation to stop, and at this point the amino acid chain coded for by the messenger RNA molecule is complete. This amino acid chain then forms the final polypeptide, and that's the end of the story which I told you that what's going on in the entire story. So what we have concluded from transcription and translation. Basically, translation begins inside the nucleus, where a particular segment of the DNA located on one strand should be copied by the process of transcription, and the end product is only one single strand of the messenger RNA having a specific sequence of nucleotide called codon. These codon have the same genetic information as on the gene of the DNA. Then, in the process of translation, this single piece of RNA molecule having the genetic information will be arrive on the ribosomal smaller subunit and will bind to its and will bind to the smaller subunit of ribosome by attaching its fifth side, and the remaining part will be outside. When it's bind, it's the stimulus for the arrival of the larger subunit. It will arrive on the smaller subunit, sandwiching the messenger RNA molecule in between them. Now, the active sites on the larger subunit are the P and A sites. The ribosome read out the sequence of codon on the messenger RNA, and according to the specific base pairing rule, each tRNA arrives. That the tRNA have an anticodon on the terminal side, and this terminal anticodon side should be complementary to the codon on the messenger RNA. So when it's arrived on the messenger RNA, it also attract the corresponding amino acid on its CCA side, forming amino acyl bond. The same process remains. Peptide bond formation between the two consecutive tRNA molecules at the ribosomal unit also join together. The chain process remains continue. The process begins from the start code and it's end up at the last with the stop codes. When the stop code is arrived, both the hydrogen bond and the amino acyl bond will be broken down. The tRNA will be released. The two smaller units of ribosome will be separated from one another, and a polypeptide chain will be released. This is the process of translation. In the next video, we will further discuss some more detail about the translation and some types of mutations. So stay with us. See you in the next video.